Hello, so today I've decided to do my presentation on Chapter 7. This is viruses and prions, and the reason I chose to do viruses is because their unique structure and their diversity compared to other microorganisms makes it really interesting to study their characteristics and behaviors. Okay, so first off, we have viruses, so they are extremely abundant. Ocean water has 10 million in a single milliliter, and lake water can have 250 million in a single milliliter. And their structures are extremely complex. So compared to a bacteria, they are 10 times smaller. They're extremely tiny, and they are found abundantly in numerous places. Okay, so viruses share similar characteristics. So as stated before, each virus, they might have a different structure because they vary, but they do share some characteristics. So they cannot reproduce unless they have a host cell, they cannot carry out metabolism, and they cannot create their own DNA. So these three points, these are characteristics of a non-living organism. To be considered living, they have to be able to reproduce on their own and carry out their own metabolism and create their own DNA. But since viruses do not fall into that category, they consider them non-living. So here's a picture of a virus. Like I said, each virus is completely different in structure, but I thought this was an interesting picture to kind of tell how complex their structures are. So the next slide I put on here is the virome. So this means the total amount of virus in your body. All cells are capable of being a virus's host, and sometimes they are capable of being in cells in a dormant state. So if they're in cells in a dormant state, usually they can stay in there for years and eventually show themselves. But typically we don't realize that they're in the dormant state until they reveal themselves. So I just have a picture of a virus here. It kind of differs from the other picture and it kind of explains how different they look. Okay, so I just put a kind of a table here to help you understand what's in the virome. So you have your endogenous retroviruses. These are the autonomous and non-autonomous. Your eukaryotic viruses. These are persistent or chronic infections, acute infections, and a novel. And you have your phages, so your lysogenic, your lytic, and your novel. And these all make up your virome. Okay, so then you have your bacteriophages. This is all of the viruses that are infecting bacteria, a part of the microbiome. So 10 to 15 power viruses are a part of the microbiome. If you didn't know, that's a huge number. And it kind of tells how much viruses are in the microbiome. And I put a little chart here. So you have your bacteriophage. This is kind of the steps that to the infection of the bacteriophage. This is how it kind of gets infected in that cell DNA. So you have your bacteriophage. It kind of lands on that host DNA. Host bacterial cell, sorry. And you can see it's infiltrating, and then it replicates, and then new bacteriophages form, and then they eventually exit. So I think it's really interesting to realize and comprehend how small the viruses actually are. So they are so much smaller than a bacterium. 2,000 viruses can fit into a singular bacterium. Um, and as you know, bacterium are pretty small as they are. Um, this is 50 million polioviruses can be found inside a regular. I'm not sure what that says behind there, but 50 million is a lot. Wherever it's fitting, it's a lot. Um, but it kind of helps you to understand and grasp how small they actually are and how dangerous. Okay, so next you have your virus components. So you have your capsid. This is a shell that surrounds nucleic acid in the central core. You have your nucleocapsid. This is a capsid and nucleic acid together. Then you have your naked viruses. So viruses that only have a nucleocapsid are considered naked. All viruses contain spikes that are proteins on the outside which allow them to dock to their host cells. So in the virus that I showed you a few slides before, you could see the spikes that were protruding from that virus. And that helps them to attach to their host cells. And also, they have that protein specific um, on the outside of their spikes so they can attach to the right cell. Okay, so then you have your disease prevention. So envelope viruses are inactivated by alcohols, whereas naked is resistant. So it's kind of all right. So now you have your viruses classification. So it is very difficult for scientists to classify them because they're so complex. So you have your order, you have your family, you have your subfamily, you have your genus and your species, and your related diseases. So there's a lot to go over in this table, but you have some of the common ones. So you have your flu, your influenza A, your hepatitis, your papilloma, some common ones you see, rotavirus, but some viruses that you can get uh, um, can be a precursor to cancer. So it's kind of mind-blowing how these tiny viruses can cause cancer as such a big disease. So I think that's pretty interesting. All right, so this is how viruses multiply. So they have different steps. So they have absorption, penetration, uncoding, synthesis, assembly, and release. Um, so I just put a chart here to kind of demonstrate how they uh, multiply. So as you can see, influenza virus becomes attached to a target epithelial cell. The coat engulfs the virus by endocytosis. Viral contents are released, and then the new viral particles are made and released into the extracellular fluid, and then the viral mRNA is used to make viral proteins. Okay, so more bacteriophages. They contain double-stranded DNA. Bacterial cells are eaten by these. At least every species of bacteria has a host, and they make it more pathogenic for humans. Okay, so then you have your provirus. The cell harbors the virus. It's an indication of persistent infections. Virus DNA is incorporated into the DNA of the host. Rosella is passed down from this virus and can be hidden in brain cells, causing lifelong damage. So that's relates back to the term before that cell viruses can be hidden in cells for a long period of time and cause those damages. So, all right. So then you have your chronic latent state. These are periodically reactivated cells for many years. An example is herpes. So the virus doesn't really go away. It's just, you know how you have flare-ups and exacerbations, but herpes is a good example of the disease. Okay, so then you have your damage to the host cell. So you have your cytopathic effects. This is virus-induced damage to the cell that completely changes its appearance and characteristics. So I put a table here. So you have your paramyxovirus. I don't know how to spell that. Cytopathic effects is faint basophilic cytoplasmic inclusion bodies, and as you can see, it completely changes that. You have your poxvirus, the pink eosinophilic cytoplasmic inclusion bodies, and cell swelling. Then you have your herpes virus, cytoplasmic stranding, and nuclear inclusion bodies, dashed arrow right there. And then you have your adenovirus, cell enlargement, rounding, and destructive grape-like clusters. So that kind of goes to show how much viruses can change the look of those cells. Okay, so then you have your identifying animal viruses. So you have your live animal inoculation is an animal that is directly exposed to those viruses. You have your bird embryos injected through the cell, great site. So this is a common injection practice, just because the bird embryo is a perfect atmosphere for those viruses to thrive. And then you have your cell tissue. So you have your culture techniques. So you have your population of viruses in that culture dish of tissues. So you have your viruses in human health. So you have COVID, warts, cold, etc. So warts are a virus, and they are spreadable. You have your Ebola and rabies have high mortality rates. So these are highly deadly infections. Some people can have long-term effects such as polio. It relates back to the brain disease, and antibiotics do not work on viruses. So I think that's pretty interesting. So then you have your vaccines. These vaccines are mainly targeted at viral diseases. The nature of viruses makes it hard to treat them, and they can cause severe side effects. Some viruses can cause cancers, as stated before. So moving from viruses, you have your prions. These are another non-cellular infectious agent. They are implemented in chronic diseases in both humans and animals. These diseases are called spongiform encephalopathies because the brain tissue can resemble a sponge. So that's kind of creepy if you think about it. It usually takes years before the disease can be diagnosed, and early signs are mental confusion and motor control. Okay, so another disease is the Creutzfeldt-Jakob disease. So this disease affects the nervous system, and it usually almost always ends in death. It is transmissible. So, okay, so then you have your mad cow disease. This is the disease in the early 1980s and the 1990s. It can be acquired from humans from direct consumption of beef. So then you have your prions. They're composed of protein. They have no nucleic acid, which confuses scientists on how they can be replicated without nucleic acid. Nucleic acid is a major component of how replication occurs. So, So more about prions. Multiple system atrophy is caused by prions in a recent discovery. They have also been studied to possibly cause dementia and other diseases. So that's interesting. So your satellite viruses, these are unique diseases that are dependent on other viruses for replication. So an example of satellite viruses is an adeno-associated virus, and it can infect cells that are also infected with the other viruses or had their DNA disrupted. Okay, so another example is a delta agent. It is a naked circle of RNA expressed in the presence of Hep B virus. It can worsen liver damage. So then you have your viroids. These can infect plants, they're extremely small, and they're naked strands of RNA. They're lacking a capsid and other coating. They can infect many important plants such as tomatoes and cucumbers.